Hello and welcome to Biology 8470, Understanding Microbiology in the spring semester. And in this chapter 1, we are going to talk about the evolution of the microorganisms and the microbiology. So the learning outcome of the first chapter that we are going to see in the 1.1 is the members of the microbial world. We are going to define the term microbiology, explain the Carl Uzi's contribution in establishing the 3-domain system for classifying cellular life. We're going to determine the type of microbe, whether it's a bacteria, fungus, or given a description of a newly discovered microorganisms, we will be able to place them in a particular group where they belong to, and we are going to provide some of the examples that how they actually do benefit us. So the question is that we all talk about microorganisms. They actually do harm us a lot. They can cause disease and different things, but they do actually have some beneficial effects, and some of them actually do not interact with us at all. They are completely free living. We're going to talk about these things as well. So when we are going to do this, that we are going to see the importance of the microorganisms. They are the most populous and a diverse group of microorganisms on the earth, and they outnumber any animals and the plant lives, but they are microscopic. Many of them are microscopic. Few of them are larger that we can see in the naked eye, but they outnumber any other organisms on the planet anywhere. Another important thing is that they are found everywhere on the earth. They are ubiquitous. Again, the word is important. The ubiquitous, that means they are found in the polar ice caps. They are found on the Mount Everest or the tall iced mountain. They are found in the hot desert. They are found on the ocean floor, where you actually do have these hydrothermal vents, there is a crack and some hot lava or the magma is oozing out. The water is boiling, and in this steaming water, boiling water condition, they will actually grow. Many of them actually do grow in hot springs near boiling mud springs. If you just go to Yosemite, you will actually find many of these, and in the diverse conditions, they are found everywhere where no other animal life or plant life actually do exist. They actually play a major role in recycling the essential elements in our world and make the place sustainable. That's why they are immensely important, and they are source of nutrients for many of the food chain. So they're extremely important to maintain the ecological balance of this earth as well. Many of them actually do carry out photosynthesis, and they are responsible for these microorganisms. A group of microorganisms we're going to talk about, which is algae and the cyanobacteria together, is responsible for production of 50% of the oxygen in the atmosphere. So very important. They also benefit in the society in the production of food. We all know that how in the beverages like alcohol industries, bread, cheese making, there are so many different fermented foods out there which are actually utilize the microorganisms. Along with that, they are the source of different antibiotics. We actually nowadays do not get the antibiotics directly from the microorganisms, but they were all discovered in microorganisms, and we actually got the recipe from them, and we actually do manufacture them in the laboratories. That's why we call them as a semi-synthetic, not completely synthetic, semi-synthetic antibiotic. All the source were actually traced back to some microorganisms, and they are also source for vitamins as well. And unfortunately, why I say that unfortunately, there is a small number of microorganisms actually cause disease in the people, in the plants, or in the animals. And that number is actually really small. That's why I say, unfortunately, although we label the microorganisms as the germs, they cause disease, but it is actually not true. Many of them are beneficial or do not interact with us at all, but actually benefit our planet in different ways. But unfortunately, a small number cause different disease as well. So when we actually see this, that in the microbiology, in this term, that again, let's define the term microbiology. The microbiology is the study of organisms which are usually microscopic in size. But again, I'm just going to contradict that word again. Microbiology actually do encompass some of these organisms which are macroscopic. So in general, These are organisms and acellular entities too small to be seen clearly by the unaided eye. This is one of the reason that's how we actually define the term microbiology, or the organisms that fall under the microbiology, generally less than a millimeter, sometimes very, very small, but some of them are microscopic. And we are going to explain this. What do we mean by this? These organisms, this is a general microbial organisms. They are relatively simple in their construction and lack differentiated tissues, except that we are going to see that there are some of them which may actually also have exceptions. So let's take a look at this, that if we all know that, say for an example, just from our regular talk, the bacteria, viruses are very small to be seen under the naked eye. But under the microbiology, we actually do study the algae, which you can actually see that when they are actually in multicellular form, we can actually see them. The fungus, or the study of fungus or fungi in plural, are also they fall under the microbiology, and many of them, like mushroom and the toadstools, they are macroscopic. Many of them are very visible with the naked eye. There are some protists which are single cellular sometimes, but some of the molds which are also protists, they are not. These molds are not fungus, but particularly the slime molds and the water molds. But when they are multicellular form, we can easily visualize them. We're going to talk about that. And the worms, or the helminths, there are many parasitic worms, and there are many free-living worms as well. This with the of the particular group of animal which you call as a platyhelminths are one of them. These are often macroscopic as well, which you can see with the naked eye. Some of them could be 10 inches long as well. So they are also discussed under the macroscopic microscopic member of the microbial family as well. So we're going to talk about that. So now let's take a look at, just bear with me with the mouse, the division and the type of the microbes. So the organisms of the biological entities are studied by the microbiologist, can be right here in a cellular form. What do you mean by that? They do have a cellular structure. What it means by that we already learned that they have to have a plasma membrane which actually will encompass some kind of cytoplasm. So they has to have a plasma membrane encompassing a cytoplasm. Now, if we just look at these cellular entities, it could be the fungi, yeast, different types of molds, and they are actually pretty well developed. They are actually, most of them fall under a group which are pretty well developed cellular structure. Then there are some which are called the protists. These are single cellular, particularly say the algae, protozoa. These are single cellular, but they often aggregate in multicellular forms. And at that stage, we actually can visualize them as an aggregate. And they also do have a pretty well developed cellular structures. Whereas the bacteria, say for an example, E. coli, Escherichia coli, E. coli, we talk about that all the time. There are many different types of them. These are all, on the other hand, do have a primitive cellular structure. We are going to talk about that. And the archaea is another group of organisms. These are methanogens, means that many of these actually do produce the methane gas, or the natural gas, and they also have a very primitive type of cellular structures. So the archaea, bacteria, protists, and the fungi, they are the cellular entities which which these microorganisms we actually do find that they do have either a very primitive type or a very well-developed type of cellular structures. But often they actually don't have an extremely well-developed differentiated tissue like in higher organisms, and that's one of the common thing that they have. On the other hand, if we have an acellular structure, means that they actually don't have a living entity. They actually, what do you mean by the living entity? There are two things that we are going to define here. First of all, they do not have a typical cellular structure. They do not have a plasma membrane. They do not actually encompass a protoplasm or a cytoplasm surrounded by a plasma membrane. That's one of the main thing. And the second thing is that they actually do not have any entity without a living cell. They actually do not have any living entity. What we mean by that, that's the viruses, viroids, or these satellites, or even the prions, to see their effect, or they can only actually cause some problems inside a living cell. So they are, in general, these viruses, viroids, and satellites, mainly these are the thing. We call them as obligate, again I'm repeating, obligate intracellular parasites. What we mean by that, once they are inside a cell, they actually do have the ability to replicate and continue their generation. But outside the living cell, they are non-living. That's why they are acellular in nature, and they're all non-living. So the viruses actually do contain the proteins and the nucleic acid, but outside a living cell, this protein and the nucleic acid will not be able to propagate them, and they do not have any living entity outside a living cell. That's why they are just as like a salt granule or a sugar molecule or a granule of sand outside a living tissue. Once they get into an animal cell or a plant cell, they will actually take over the cell and control the cellular machinery to make progenies. So that's why we call them as obligate intracellular parasites. The viroids, on the other hand, are composed of completely of RNA, and these RNAs actually infect plants, mainly the plant entities, and these are replicating RNAs that we are going to talk about. So these are also, they cannot actually have any kind of function outside a living cell. They need to be inside a plant cell. They particularly infect the plants. On the other hand, these satellites, these are actually nucleic acid encased in a protein cells. Why we call them satellites and not viruses? You can see that they are also protein and the nucleic acid. The satellites actually only replicate when they do have a helper virus infected the same cell. And repeating, a cell infected with a virus can have some satellites. Now, not all viruses actually have satellites. The particular satellites actually require a particular type of a virus replicating in that cell, and they can actually have that. Just giving an example, say for example, adenovirus. Adenovirus actually can cause cold-like symptoms as well. It's an animal virus. When an adenovirus actually is replicating in a cell, it can have some satellites which are nucleic acid and enclosed in a protein cell growing in there. Sometimes it is implicated that these satellites, which only can grow in presence of a virus growing in that cell, can complicate the outcome of the disease or actually can increase the severity of the disease. We'll talk it somewhere else. On the other hand, this prion is very different. Here we're talking about viruses, protein, nucleic acid, viroids, only nucleic acid, RNA, only in the plants, satellites, nucleic acid, and proteins. Prions are proteins only, and these prion proteins are present in our normal cell, and somehow these proteins actually get misfolded in the cell and become an infectious protein. What we mean by that, once they actually get misfolded, this can be transmitted through the meat from one animal to the other. Say for example, if we just have a misfolded prion protein in a cow or a beef, we can just probably, if we just consume that meat, we can actually have this protein from them. Now the question is that you will immediately ask that if we just cook the meat, what is the problem? Unfortunately, that's the problem, that the cooking process actually do not destroy these prion proteins. They can survive autoclaving, that we are going to discuss somewhere else, which is a pressurized steam at about 15 pound per square inch pressure and about 121 degrees Celsius, which is way more, 21 degree more than a boiling water temperature. Even in that condition, the proteins cannot be destroyed. So these are really some deadly proteins which can actually go into a neural cell, and they can convert this neural cell into they can kill these cells and unfortunately can convert your normal prion proteins in a cell, in a neuron cell, into infectious proteins. So a prion can convert a disease prion protein can convert your normal prion protein into infectious prion protein. So right now it's not needed, just giving an overview of that. We have a separate chapter that we are going to talk about that in the later part. So now we are going to talk about the types of microbial cell. The prokaryotic cell lack a true membrane delimited nucleus, and this is not absolute. So what we mean by this, that a prokaryotic cell is a primitive type of cell, primitive, and the word comes from carrion. Carrion means a nucleus. So pro means primitive nucleus. So if there is a very crude definition is that if you have a cell where the nuclear material is not enclosed within the nuclear membrane and the nuclear material is scattered within the cytoplasm, that's a prokaryotic cell. But this definition is not correct. There are many members of these prokaryotes which actually do have some internal membranes. They have compartments, and there are many different characteristics which actually are different, and that's the reason that we are going to see that at the beginning of the next chapter that some of the scientists actually challenged this process. And in 2006 a Nature paper came out where one of the scientists actually proposed that this prokaryote term is very controversial and should be discontinued. It is not. Some of the scientists agreed, some of them actually did not, but it is definitely very controversial at this point, and we're going to talk about that more in detail in the next few chapters. On the other hand, EU means true. EU means true, and karyote is a nucleus. So eukaryotic means that it is a true nucleus. These cells have a membrane enclosed nucleus, more complex morphologically, and usually much larger than a prokaryotic cell. So say for example, our cell, the human beings, we actually are eukaryotes, and there are fungus or even the protists, they actually do have eukaryotic cells, whereas the archaea and the bacteria that we talked about before actually are within the prokaryotic type of cells that we are going to talk about. So the next that what happened, that classification scheme, that how we actually put all these different organisms in the world into different domains or classify them. Now, historically, before 1970s, in the 1950s, 60s, there were actually 5 different domains. We actually have the plants, we have the animals, we have a kingdom called Monera, and there are different types of fungus, and depending upon how they look like, the scientists actually classified them in many different ways. There were many arguments about that, but they were as many as different kingdoms as such. But in 1970s, Carl Uzi and the Fox, they actually discovered a particular way to classify organisms. What they found that the ribosomal RNA genes, what do we mean by this? First of all, let's dissect this word. The ribosomes are the protein synthesis machinery that is found in all organisms. They're found in prokaryotes that we talked about. They are found in eukaryotes, and they are made up of ribosomal RNA and the protein. These ribosomal RNA actually are coded by ribosomal RNA genes, which are encoded into the DNA. Again, and repeating, ribosomal RNAs are the building block of the ribosomes along with the protein. And these ribosomal RNAs are actually transcribed from the genes which are in the DNA. And these sequences of the ribosomal genes actually are very, very conserved. That means they are extremely similar in between the organisms. And when we actually look at their sequences of these ribosomal genes, we actually found, or the Carl Uzi Fox actually found, that there are actually not technically different animal kingdoms or plant kingdoms or kingdom Monera or kingdom fungi. They are not that varied. On the other hand, there are mainly three domains. One is a bacteria, true bacteria. Another is archaea, and another is eukarya or the eukaryotes. So they actually found that we actually had a common ancestors, and from these common ancestors, after origin of life, the bacteria actually developed independently, and the eukarya and the archaea actually did also have a common origin, and they diverted into the archaea, and they were divided into different groups, and the eukarya were different into different groups. So this was a complete shift in the understanding that how we classify the microorganisms. And you can actually see the fungi, the animals, algae, plants, and we are just next to each other in relationship. Earlier, these were all put into different kingdoms, and they were very distant as such, but you can see that they are very closely related, whereas the bacteria is farther from the archaea as well. So these are the three different domains of life, and this is the modern classification which is based on Carl Uzi Fox's ribosomal RNA gene sequence alignment, and this is basically the molecular basis of classification which is followed today. So now we are going to differentiate that the domain bacteria and the domain archaea, why they are different. If you actually look at them, both are microscopic, and classically they are prokaryotes lacking a true nucleus, and you may think that they are very similar, but they are distinctly different, and as evidence from the ribosomal RNA sequence. So the similarity, they are usually single-celled, and they distinguish from the other. They are all single-celled, but they distinguish from the bacteria by their unique ribosomal RNA gene sequences. This is the main difference. Majority of the bacteria actually do have something which is called peptidoglycan, also known popularly as a murine. So the peptidoglycan, if you dissect the term, is peptide, means they have some proteins in there, and glycan means carbohydrate. So these are some of the carbohydrates attached with amino acids. Peptidoglycan, a very typical type, they are found in the bacteria. And this is one of the major differences, and these could be all your quiz question, exam questions. The archaea actually lack the peptidoglycan. On the other hand, most of them, bacteria actually lack a membrane-bound nucleus. That is also true for them as well. On the other hand, the archaea actually do have very unique membrane lipids, and we are going to see that these membrane lipids are actually some of the molecular evidences that scientists actually look for when they are looking at the molecular fossils, and they actually try to trace back the origin of life depending upon that. So we're going to talk about that in a moment. Another thing, the bacteria are ubiquitous. Some live in extreme environments. On the other hand, if you can see that archaea, many of them actually live in extreme environments. What do you mean by the extreme environments? They are found in hot water springs. They are found in these boiling boiling ponds with muds and all those. They are found near the hydrothermal vents near the ocean, when you have some of the cracks in the crust of the earth. Lava is oozing out, the water is boiling. You do have archaea in those areas as well. They also are found in extreme cold conditions as well. On the other hand, you can see that cyanobacteria actually produce a significant amount of oxygen, along with in combination with some of the algae, which are the eukaryotes as well. So these are some of the characteristics of the domain bacteria and some of the domain archaea, and they are distinctly different, and this was established from the ribosomal RNA sequences. So on the domain eukarya, first what we have is the protist, and the protists are generally larger than the bacteria and the archaea, and they actually do have a membrane-bound nucleus, and they do have different types of organelles. They do have different functions for these organelles. We're going to talk about that. But they are all single cells, or at least one part of their life cycle, they are single cells, and these are called the protists. So what are the different types? Algae that we can actually see, these are photosynthetic organisms, and if you actually look at a single cell algae, they are microscopic, you cannot see them. But when they are numerous of these algae, they are in a multicellular condition, you can actually visualize them. Say for an example, if you're just looking at some water in an aquarium, sometimes this water seems to be green, and why it is? Because there are some algal blooms in there, and these algae, you cannot see them individually with the naked eye because they're a single cell, but the aggregation gives that the green color, and from that you can tell that they are photosynthetic as well. On the other hand, the protozoa are maybe motile. Sometimes they are non-motile as well. They are hunters, sometimes they are grazers, means that many of them actually are motile. They look for food and they graze upon food, but some of them actually don't. And some of these protozoa can be actually disease causing in the human and the animals as well. And they are actually a very well developed cells as well. Say for example, an amoeba is a protozoa that is the regular amoeba will not cause any disease, but there are some of the species or a different species like a different genus like an entamoeba, which can cause severe gastroenteritis as well. So there are different free living form causing not any harm, or there are some disease causing protozoan as well. The slime molds and the water molds are two interesting types, and why we call them molds or fungus, because once they were thought to be fungus, but they are not. So the slime molds actually have two life cycle stages. So they actually do have a single cellular life cycle stage, and that's why they are under the protist, and they have a multicellular stage, and that's the reason that they were thought to be fungus as well. And you will be able to see these slime molds in many different areas. Some of them are very brightly colors. They are found on the rotten woods and many of the other areas you have seen them. And these are sometimes called as a naked or moving protoplast as well. Very interesting characteristic. We're going to talk about that somewhere else. On the other hand, the water molds are actually also have two different varieties, that they are also have a very fuzzy structure. They have a single cellular form, multicellular form, and they're once thought to be the fungus, but they are not. And there is something related to this disease, that they are devastating disease in the plants. And you all know that there was a great the famine in Ireland, which actually caused a mass exodus from the Ireland, and they all came to the United States in the 1840s. And these famine were caused by disease in the potatoes. They were just all rotten, and they were caused by this the potato blight disease, which is caused by a water mold. They actually they not necessarily they do not need to be in water. They can actually grow in moist soil or where there is a high water content as well, and can cause severe diseases in plants as well. On the other hand, the fungi can exist in two different forms, and we are going to see that we actually use a word which is called a dimorphism. Morphics means structure, di means two, dimorphism. The fungi is plural, fungus singular, can actually be in two different forms. So what are these different forms? They can be in an unicellular forms. We actually call them commonly as yeast, very, very common. Brewer's yeast, we all the time use that. We are used in the baking industries as well. These are unicellular form. On the other hand, they can also exist in a form which we call as a multicellular form. They are often filamentous in structure, and we call them as molds or mushrooms that we actually see. They are multicellular forms. So here, the words molds actually make sure that it doesn't distract you that these are slime molds and the water molds are protists, but they are not fungus. So the next that we are looking at, that some of the acellular infectious agent, for example, viruses, smallest of all microbes, requires a host cell to replicate, cause a range of disease and some cancers as well. And we're going to talk about a complete separate chapter. And we talked about the viroids as well. These are infectious agent composed of RNA, and they are mainly infectious to plants only. On the other hand, the satellites are nucleic acid enclosed in a protein cell, very much like the viruses, but most co-infect a host cell with a virus to complete their life cycle, as we just discussed. They cannot replicate on their own. On the other hand, the prions are the infectious protein, and we are going to talk about them along with the viruses in the same chapter in our module 1 towards the end of the module 1. So the section 1.2 deals with the microbes have evolved and diversified over the billion years. And we are going to just take a look that propose a timeline of the origin, design a set of experiments that could be used to place a newly discovered cellular microbe on a phylogenetic tree based on a small subunit of the ribosomal RNA sequences. We are going to dissect that word as well. And we're going to compare and contrast the definition of the plant and animal species, microbial species, and some of the microbial strains as well. Some definitions there. So when we are looking at the origin of life, and some of you who have accessed the PowerPoint slides, this is just a correction that here you will going to see that the origin of life, and after this slide, there was another slide, and this is, if you have downloaded the PowerPoint, these two slides will be different. These slides did not have the correct content, so I have changed this. This is just a reminder for that. So the origins of life actually is controversial in the sense that we actually exactly do not know that how they arose, because we do not have any direct evidence. So we actually have to have indirect evidences to have some theories about the origin of the life. So what we try to do, or the scientists trying to do, that they all try to find ancient characteristics in extant microorganisms. What we mean by the extant, that they are which are present today. Extinct means they are gone. Extant means they are present. So there are some characteristics which are extremely primitive, and we try to find them, that whether these are actually clues which can go into an indirect evidence for the origin of life. And we are going to talk about that in a second. And then also we can actually look at the microbial fossil record, but many of the scientists actually look at this as a skepticism. Say, for an example, this Archean Apex shirt, this is actually a classic example, and this is the picture of that, and that you also will find in the book. So this was considered as a microbial fossils, and the scientists actually proposed a theory that this is actually some archaic type of microorganisms as such. But this was actually proved to be completely wrong, because scientists actually discovered later, or proposed a theory, that this type of structure was formed completely by a geological cycle. This is like a chemical reaction, geological reaction over the period of time, which actually gave a fossil-like structure, where the scientists actually thought that this is some type of archaic microorganisms. So that's the reason that the microbial fossil records are actually not very reliable. On the other hand, this is actually a very reliable way to look into it. We actually look at some of the molecular fossils. So say, for an example, a classic example is a Hopanes. This Hopanes is actually a group. These are bacterial lipids, and this is a typical structure. You can see that these are interconnected cyclic compounds, and this is a Hopane molecule. And some of these molecules are actually found in the bacterial plasma membrane. So the scientists actually did find some of these Hopane-like molecules in the ancient structures, and they can actually relate that maybe that these are some of the links towards the origin of life. So these are some of the ways that the scientists actually draw the attention towards the indirect evidences and argue about that how we can actually think about the origin of life from these evidences. So here we are just looking at an overview of the life history of Earth as well, and I don't want you to memorize all those. And you can see that probably the Earth probably almost like 4.5 billion years old, and about 3.8 to 3.5 billion years after that, probably the first ancestral type of cell probably actually arose over the period of times. And we actually think that about 2.5 to 2 billion years ago, the first eukaryotic cells actually developed. And it took a huge amount of time over the period of time to get into the reptiles or the amphibians or the dinosaurs. And these are pretty recent as such. You can think that the dinosaurs existed probably 65, 70 million years ago, which in the time scale is actually a pretty recent event. So this is just, again, overview of that and read about, but we don't ask you to memorize any of these time scales, and we're going to not ask you, quiz you on this, or ask any of these timelines. So here we are actually going to have an idea which actually do have the relationship of DNA, RNA, and a protein in the cell. And sometimes we used to call this as a central dogma, that how the DNA, RNA, and the proteins are related to that. So this is a modified type of a central dogma that we are going to explain. So first, let's explain that what was the earlier central dogma that we actually know about. So the earlier central dogma is that your hereditary material is the DNA that is changed into the language of the RNA, which is transcribed into the language of RNA. And then from the RNA, it is translated into the protein to the different functions. So from DNA, RNA, protein, the information flow is this way across all living organisms, and that used to be called as a central dogma. But in reality, the process is way more complicated into that, complicated compared to that. So let's have a look at what it is. So here the DNA serves as a template for new DNA synthesis. So the DNA can replicate. So this actually stores all the genetic information. So the DNA is the storehouse of the genetic information. Now what happens, that follow this line carefully, it stores the genetic information and it encodes sequences of the nucleotides in the RNA. This process is called the transcription, encodes the sequence and nucleotides in the RNA. Once this transcription is done, this RNA is actually read by the ribosomes, and this functions in the synthesis of a protein by the help of a ribosome. So the information flow is from DNA, which is stores the genetic material, to RNA, to protein. It still pretty much follows the central dogma. But let's take a look that there are some feedback loops in there and how this actually works. So from the genetic information, you can see that the genetic information encodes the sequences of amino acid in the protein, but they cannot directly go into making the protein. It has to go through the RNA. Now, once the RNA actually makes the protein, this protein actually do have many different functions, which is upstream. So let's take a look one at a time. So once the protein is produced, these proteins can be enzymes, which actually do different functions. One of them catalyzes the synthesis of DNA. So the protein, giving an example, can form a DNA polymerases, which will actually synthesize the DNA. The protein will catalyze the synthesis of RNA. So the RNA polymerases will synthesize the RNA. So from DNA, when you are looking at, serves as a template for synthesis of new. To make new DNA, you need the protein. To make new RNA, you need new proteins. And along with that, they are involved of synthesis of more proteins, because the ribosome actually do need proteins along with the ribosomal RNA. So the proteins are needed for their own synthesis as well. Along with that, they carry out a lot of chemical reactions. We call this as a metabolism. All the sum of the sum of all chemical reactions in the body. There are many of these reactions. Say, for an example, think about that glycolysis, Krebs cycle, or the digestive process. All these chemical reactions are done by these different enzymes. Along with that, they also do have some structural function as well. If you actually look at these particular functions, catalyzing the DNA synthesis, catalyzing RNA synthesis, helping in more protein synthesis, carrying out chemical reaction, or also regulating the expression of genetic information. What do we mean by this? Take a look at this, that once these proteins are actually synthesized, they can control what kind of genetic information is going to be produced into the protein and what not. And this is the regulation of the gene expression. You have seen that in a different chapter, maybe in cell biology, where you can think about this way, that a cell, particularly a liver cell, which is produce insulin. So the proteins in that cell actually regulates or tells the cell to produce insulin out of the liver cell. But on the other hand, say in a nerve cell, the proteins will actually not tell the cell or the neurons not to produce insulin. The genetic information in a neuron cell of yours or the human beings, genetic information in a neuron cell or in a neuron or in a hepatic cell or a liver cell is exactly the same. Both contain the exact same DNA. But one cell is producing insulin and another is not producing. And this is how that all different tissues are producing different proteins, different enzyme, different structural proteins. And these are all controlled by the regulation of gene expression, which is essentially the protein controlling that process. So that's the process. On the other hand, these are all the functional part of the proteins. So the next is the structures. There are many different structures that are produced by the protein. Say, for an example, the collagen fibers, which actually the elastic fibers, reticular fibers, these are the different structural proteins that are present and give the animals and the plants different forms. Say, for an example, our hair or nails, these are made up of keratin. These are all proteins, very, very hard proteins. So from here, let's take a philosophical note from here that we have seen that we have a DNA, RNA, and the protein. So the only information that is stored as a genetic information in the DNA is to make different proteins. So the question is, so what happens to carbohydrate and the lipids? And that's the interesting part, that the proteins are the different enzymes, and these enzymes are actually can catalyze the formation of different lipids or carbohydrates, or they can actually obtain them in terms of food from the environment and process them. So they can obtain or synthesize proteins and the carbohydrates with the help of different enzymes. So protein is the only thing that it needed to be coded and synthesized by this process. So this is basically the overall cellular dogma that is very important and will be required in the some later part of the chapter as well. So the earliest molecules, or the RNAs, that what we are going to talk about here, that some RNA molecules are actually thought to be more ancient, and probably the original molecule probably were RNAs. The reason for that, the ribozymes are some of these molecules which are actually can cleave or actually can undergo chemical reactions. And these actually RNA molecules can form actually peptide bonds, perform cellular work, and their replication. So the scientists actually thought that the earlier cells may be RNA surrounded by the liposomes. So the liposomes is just like a lipid membrane, which enclosed some of these RNAs. And these RNAs actually were able to synthesize different proteins. And at the later part, the DNAs actually developed, because RNAs are single-stranded and they are actually not much stable, whereas the DNA genomes are much more robust and stable. And that's why we think that the RNAs actually evolved first, and then the DNAs evolved at much later period of time. So today's version, the cellular pool of RNA in modern day cells exist and is associated with a ribosome, like a ribosomal RNA, transfer RNA, which brings the amino acids to the site of protein synthesis, or the messenger RNA, which is the encoded nucleotide sequence in the DNA transcribed into the form of mRNA. And this actually tells which amino acid is going to be attached next to which amino acid in a protein sequence. So these are the main three types of RNA. There are some other type of RNA that you have heard about, the microRNAs, and there are some other types of RNAs that we are not going to discuss right now. So the RNAs are catalytic in protein synthesis, and RNA may be precursor of the double-stranded DNA. And that this is the theory that is universally accepted as of now as well. Interestingly, one of this adenosine triphosphate is essentially a nucleotide. It is a ribonucleotide. So what is the difference? That in the DNA, you have a deoxyadenosine triphosphate, whereas the adenosine triphosphate is the nucleotide which is present in the RNA itself, and it is the energy currency or source of energy in the cell. So this is a very interesting idea that we all know, but it is a nucleotide of the RNA. RNA also can regulate the gene expression. What do you mean by that? We just talked about that the proteins can regulate the gene expression, which will be, what do you mean by that? Whether a protein will be produced in the cell or not is the gene expression. This can be controlled by the proteins we just saw, but RNAs can also regulate whether the gene will be expressed in a cell or not. So the next that what we are looking at, that the earliest metabolism, the early energy sources were under very harsh conditions, and the earth was at that time a very hostile place, and the main metabolism type was based on different inorganic materials. For an example, like iron sulfide. Many of these ancient cells actually utilize many of these inorganic compounds to produce many of those. And on the other hand, gradually over the period of time, photosynthesis actually evolved, and photosynthesis actually drove this planet from an anaerobic condition into an aerobic planet. And these cyanobacteria, which were the ancient bacteria, evolved about 2.7 billion years ago. And these are actually some of the first particular bacteria which were able to photosynthesize and produce oxygen. Over the period of time, they changed the atmosphere of the world into an aerobic condition. So these things that what you are seeing are called that stromatolites. These are a mineralized layer of microorganisms as such. So these are actually found in many of the different areas, and we actually can look at some of these structures and have an idea about the microbial form in the ancient world. But we have to be very careful that when we're looking at the fossil structure, as we discussed above, that there could be many aberrant things that could result from biogeochemical cycles, which is a chemical reaction or a geological reaction, but may not be actual microorganisms as such. So these are the earliest metabolism, that from inorganic metabolism using different chemicals, it actually moved into the organic photosynthesis, which means that in the presence of sunlight and the carbon dioxide, these different types of organisms started to make their own food. And this is the organic evolution in the process of earliest metabolism. Say, for example, they were able to make sugars, which were not possible. The sugars can act as a source of energy, whereas these were not available to the earlier metabolic process, whereas these inorganic compounds actually were the source of energy that is being utilized with some of these primitive organisms, where the organic evolution to produce food by the process, by harvesting the sunlight, by the process of photosynthesis, and fixing the carbon dioxide into food like carbohydrate, actually evolved from the process of the inorganic portion of inorganic part of the metabolism. So the next part that we are looking at, the evolution of the three domains of the life, and we actually have an universal phylogenetic tree, and this universal phylogenetic tree is based on the comparison of the small subunit of the ribosomal RNA. And what we mean by this small subunit of the ribosomal RNA, the ribosome, and we are going to discuss that in detail in later part, but the ribosomes actually do have a large subunit and a small subunit. And again, the large subunit can be composed of as many as 2 or 3 different types of subunits, and the small subunit can be composed of, again, 2 different types of subunit. And one of those small ribosomal subunit, ribosomal RNA sequences, can give us some clue about the evolution and the 3 domains of right. So what we actually do, that this is the process that we are going to discuss, that we align the ribosomal RNA sequences from diverse organisms and compare the differences counted to derive a value of evolutionary distance and relatedness, but not time of divergence determined by this way. So let's take a look at what we are actually looking at. That we actually take the cells from an organism 1, and then we actually lyse the cell, release the content, isolate the DNA, and we use a PCR reaction or polymerase chain reaction to amplify or purify the SSU ribosomal RNA genes, or the small subunit ribosomal RNA genes. How do we amplify? We use some of the primers that is known or conserved, or some of the sequences of these small subunit of the ribosomal genes are very conserved among organisms. So we design primers with that, and then we can actually get the full sequence of that small subunit of the ribosomal RNA genes. We sequence these genes and get these ADCGs, and this is from organism 1. We can do 2, 3, 4, 5, 6, as many as we want. And now suppose we have these different 4 organisms, 1, 2, 3, and 4, and we have aligned these sequences of the small subunit of ribosomal RNA sequences, and we are finding that these are the alignments. So if you actually look at that, we are looking at ATG, TCA, AGT. If you're just looking at these areas, if you look at these areas, what we are finding from here, that when we are looking at the different distances, that what is the conserved sequence in between 1 and 2, we find that there is about 0.61 is the corrected evolutionary distance in between them. In between 1 and 3, it's 0.30. 1 and 4, 0.44. Between 2 and 3, 0.44, same. 2 and 4, 0.44. 3 and 4 is 0.30. So from this initial evolutionary distances calculated, we have this corrected evolutionary distances. And if we just put it in the phylogenetic tree, if you can see that from 1, we have the relatedness with the 3. So we have this distance from here. Then you have a smaller distance from here. And you can see 4 and 2, they are actually computed out the different evolutionary distances. So in between 1 and 3, it is 0.23 and 0.08. And if you just actually calculate these 2 numbers, and you will just actually come up with the numbers which is pretty close to them as such. Say 0.23 and 0.08 will give you a close number to almost like close to 0.31, and that's what the distances is in between. So if we just look at the in between 3 and 4, so if we're just looking at 3 and 4, let's just add up these. So you have about 0.15, 0.08, and 0.08. So here it is 0.16 and 0.15, so about 0.31. So that's kind of a distance in between them. So this is how we actually relate these sequence and look at the different sequences, and after aligning them, just finding the evolutionary distance to place them in our evolutionary tree. So this is actually we're responsible for the Carl-Uzi-Fox's the 3 different domains, which is the bacteria, eukarya, and the archaea. And these are the 3 different domains. That is the classification of the modern organisms on Earth. So the last universal common ancestor is the root or the origin of the modern life is on bacterial branch, but the nature is actually still controversial. The archaea and the eukarya actually evolved independently of the bacteria, and so the archaea and eukarya diverge from a common ancestry. So the archaea and eukarya actually have a common ancestor as well. But the bacteria is a little bit more distant. They evolved in a different way as such, and this is actually all computed out from these ribosomal RNA sequences that we just talked about. And this is the most modern accepted molecular evolutionary guidance that we actually can think about. Another idea, so the endosymbiotic hypothesis, it basically tells that in an eukaryotic cell, how the mitochondria, chloroplast, and hydrogenosomes, these structures actually developed in an eukaryotic cell. So the hypothesis is that the mitochondria, chloroplast, and these structures were actually different types of bacteria which were engulfed by a cell, and they actually modified their functions and became a part like a mitochondria and the chloroplast. And how they actually understood that this actually really happened over the period of time, because they found that in the mitochondria there are some small subunit ribosomal RNA genes which actually show some bacterial lineages as well, like genome sequences closely related to some of the proteobacteria or a prochrolone respectively actually do have some of the mitochondria or chloroplast sequences. So prochrolone is chloroplast, proteobacteria for mitochondria. And also the hydrogenosomes are some of that we are talking about. That let me explain this, that how this actually worked. So initial ideas about the scientists that the mitochondria and the chloroplast were some of these bacteria, and they developed over the period of time. But that hypothesis now changed to what we called as a hydrogen hypothesis. So the hydrogen hypothesis, you will see it in your textbook, it is written in there, where the scientists actually think that there were some anaerobic bacteria which actually were engulfed by these of the cells, and over the period of time these bacteria actually changed into aerobic type of bacteria, and that's how the mitochondria actually developed. And then there are some of the other structures also in there, that some of the bacteria actually used hydrogen as well as carbon dioxides, which actually developed into the hydrogenosomes, and these are the anaerobic endosymbiotes. So essentially the anaerobic bacteria used the hydrogen and the carbon dioxide and developed into the hydrogenosomes, and that actually supports that those are some of the theories that the scientists actually think that how these organelles actually developed over the period of time. So the next part that we are going to talk about is that how the evolution of the cellular microbes actually happened over the period of time, and the mutation of the genetic material actually led to the selected traits over the period of time, and the new genes and the genotypes actually evolved, and these producing mosaic of genetic information. Now the reason is that why this actually happened, and over the period of time as the cell environment in the atmosphere actually changed over the period of time, many of these bacteria actually evolved due to that. So how this actually relate? So remember that the most of these ancient organisms, particularly what we call as a prokaryotes, controversially the bacteria and the archaea, they actually do not have sexual reproduction. So they actually do have a process of reproduction which we called as a binary fission, and we called as this is a horizontal transfer of genetic material, or it could be a vertical transmission of genetic material. What we mean by that one bacteria can divide into two bacteria, that is possible. Now this one bacteria can actually receive a genetic material from another bacteria as well, and receive some new genes as well. And these were all, this vertical and the horizontal gene transfers were all evolved, all involved in this evolutionary process as well. And the overall idea or the philosophical perspective is that The bacteria and the archaea, all these primitive organisms actually divide extremely fast over the period of time, and they randomly actually gathered many different mutations, and many of these mutations were lethal, of course, that they will not be able to survive. On the other hand, as the atmosphere in the earth changed, many of them were actually more adapted to the newer environment, so they were selected over the period of time and evolved, whereas the ones they were not fit actually gradually got deleted, or gradually they were unable to survive over the period of time. And what we actually think that's also an interesting thing, that there are many disease-causing organisms, and they are some of the very late evolution, because they must happen once there are different types of animals actually evolved over the period of time, and these animals needed to be in their environment, so that a genetic mutation that may have been before, but that genetic mutation to be a disease-causing organisms actually were probably not selected, because in the environment there were nothing where it can actually replicate. So they were selected when the organisms like a plant, animal, or the human beings were there. So these are some of the overview of the evolution of that. And bacteria and archaea increase the genetic pool by the horizontal gene transfer within the same generation. That we mean by this, that bacteria divides 1 into 2, 2 into 4, and in the same generation they can actually transfer genes by many different methods. So say, for an example, we're going to talk about that, like a conjugation or the transformation. These are some of the method that this horizontal gene transfer can actually lead to many different types of traits as well. Giving a classic example of that we actually can see in our environment or in real life as such. Say, for an example, you can actually have an E. coli, which is very naive, doesn't cause a disease. On the other hand, you have a rogue E. coli, which actually does have a gene on a plasmid that we are going to define later in the part, in a small movable genetic element, the double-stranded DNA element. And this rogue E. coli can transfer or horizontally transfer that gene to some of the naive E. colis, and they will actually transform into a rogue E. coli. This is just like a hypothetical example of that, but many of the bacteria can do this process to evolve over the period of time to acquire new genes or new traits. So the microbial species, on the other hand, the eukaryotic microbes, fit the definition of reproducing as isolated population. Bacteria and archaea, as we said, don't reproduce sexually, and they are referred to as strains. So very different. A strain consists of a descendant of a single pure microbial culture, and they could be in a biological entity, they called bioverse, morphological difference, morphoverse, serological differences, seroverse, or pathoverse. What do we mean by that? That if we just have E. coli, a species E. coli, and we talk about this all the time, and sometimes these are in the news, that we actually do have these E. coli infection, which can lead to some of the recall of these the grocery store lettuce or some of the green vegetables as such, and cause diarrhea, some gastrointestinal problem, bloody diarrhea. These are some rogue species of E. coli, and we do not call, they're all E. coli, Escherichia coli. They have the same genus and the same species, but they actually are a different types of pathogen, either a serover or a pathover. So we call them as a strain, a different strain of the bacteria. So you can have a bacteria called a K12 strain. You can have a bacteria same with another strain. So if K12 is a common E. coli strain, you can have an E. coli strain called OP50. That's an E. coli strain. On the other hand, these pathogenic strains, which we called as the H7, which does have a different type of sugar or carbohydrate on its outer membrane different, and we actually can name them like those. So Depending upon that, these are called the strains, but not different species. On the other hand, the binomial nomenclature, we have a genus, which is all starts with a capital, and the species after that. Say for an example, if we just like Escherichia is, which is the small form of, we call this E, that is a genus, and the coli comes from the colon that is common in there. That's the species as well. And each of these genus and the species also can remain in many different bioverse, morphoverse, seroverse, or pathoverse. These are the different entities, but we do not consider them as different species. So in the section 1.3, this is actually, we are going to talk about some of these scientists over the period of time, that how they actually develop some of the new tools to study the different microbes that they were developed. We're going to talk about some of these scientists. We're going to talk about an experiment which actually will lead to some that how the scientists actually discovered the causative agent for a disease. And we are going to talk about postulates of Koch, and we're going to see that there are some cases where we cannot determine the cause of a disease, and those were some of the drawbacks of those, and we're going to discuss that as well. So here, what we actually seeing right here, that the microbiology, the study of the microorganisms, and the tools that we study the microbiology is microscopes, different culture techniques, molecular genetics, genomics. These are some of the ways that we actually study microbiology, and we will deal with these in detail in many different areas as well. So here we actually looking at some of the important events in the microbiology from 1665 to say 1911, and by any means I'm not going to ask you to memorize all these different names over the period of time that what happened as such, but just giving you an overview of an idea, so that just studying the subject, you have an overview that over the period of time that how things develop. Say for an example, right here, if we look at that, right, 1665, Hooke publishes the Micrographica, but see right here, the spontaneous generation of maggots was proposed. So the scientists actually at that time, Redi told that these can actually, and things can generate spontaneously. So these were not a proper observation because they did not have the proper tool. Right here, the Leeuwenhoek actually discovered the microscope, and that actually changed completely that how the scientists actually look at these this world. And right here, there was some attack of the spontaneous generation. See, 1798, even way before that we knew viruses, Jenner actually developed the smallpox vaccination. So this is one of the name. These are the landmarks. So please remember this one. I don't want you to remember all of them, but this is the Jenner for vaccination is a one that you want to remember. John Snow here actually traced in London, sourced a water pump as a source of cholera in London. So this was also a landmark discovery, and please remember this one as well as John Snow for the cholera. And right here, 1861, this is also another landmark discovery right here. The Pasteur actually disapproved the spontaneous generation. So remember this one right here. So on the other hand, so right here, Cox demonstrate that Bacillus anthracis causes the anthrax. That was one of the thing. And 1884, the Cox actually developed this Cox postulates. So these are some of the important ones, like a Jenner, the John Snow, Pasteur, Cox postulate. These were some of the landmarks that I want you to remember. The other ones are not that important. Pasteur, of course, developed the vaccine, but the first one is the most important ones. So here you can see 1887, the sulfur nitrifying bacteria was developed. Beijering isolates the root nodule bacteria, proves the first virus was the Beijering, proves the virus causes tobacco mosaic disease right over the period of time. So 1911, Rouse actually discovers a virus can cause cancer. Nobody believed him, gave him a Nobel Prize 6 years after that discovery as well. But here, I think that the name that I'm pointing out is Jenner, John Snow, Pasteur, and Koch, Robert Koch. So these are some of the important ones that you want to visit. So let's take a look at this. These are some of the earliest microscopic observations, and Robert Hooke, and if you remember, Lanton van Leeuwenhoek as the first person to observe microorganisms accurately. That would be great. But again, these are some of the historical perspectives and very self-explanatory as well. So these are some of the conflict over the spontaneous generation, and these are very self-explanatory. You can just read it and its historical perspective, and really I'm not going to ask every name and year. That's not the purpose of this course. So you can just read over the thing and just take that historical perspective of that opinion. And the Louis Pasteur was the one where he challenged this spontaneous generation with this famous swan neck flask experiment, and we're going to explain that. And this disproved that the spontaneous generation doesn't happen. So what the swan neck flask experiment is that Pasteur actually put a liquid in a flask, and he actually applied vigorous heat to that and boiled these. And he actually used a long neck like this, a swan neck like structure, and by drawing the glass tube over that, and then he stored that over the period of time. So what he found that when he stored this swan neck flask over the period of time, with the intact neck, airborne microbes are trapped at the base, and the broth remained sterile or clear for a very, very long time. On the other hand, in the experiment, he has shown that if you break that neck of that swan neck, where the aerobic or the aerosolic or the airborne microbes can get into the broth, over the period of time, growth occurs, the broth becomes cloudy, and it is spoiled. So this was the greatest blow to the spontaneous generation, that the microbes actually can only come from some source. They cannot spontaneously generate into a sterile broth or a sterile media. So that was the experiment. So these are some of the final blow that John Tyndall demonstrated that dust carried microorganisms, and then Ferdinand Kahn, heat-resistant bacteria could produce endospores. These are some of the historical perspectives as well. And again, I do not want you to remember each and every name, just have a historical perspective of that. So the role of microorganism in disease was actually not immediately obvious. The infectious disease believed to due to the supernatural forces and establish a connection depend on it actually took some time. So essentially what you can see that the Agostino Bassi actually showed that the disease in a silkworm is caused by a fungus. And then the potato blight in Ireland is actually caused by a water mold that we talked about before, that these are some of the indication. Smut and rust fungi cause cereal crop disease. These were some of the early evidences that between the microorganism and the diseases. And again, you do not have to remember the names of that. Just want you to have a sense that how the perspective of the scientific inquiry led to the modern discoveries of the basis of the disease. In Louis Pasteur, further early evidence of the relationship demonstrated that microorganisms carried out fermentations, helping the French wine industry, publishing papers on fermentation in this area, and developed the process of pasteurization to avoid the wine spoilage by the microbes. And these are the huge contribution of the Louis Pasteur, and we use that pasteurization technique to actually keep many of these products like a wine, beer, or milk products so that they don't get spoiled easily. So the technique is that many of these products, fermentation products or the milk products, if you actually vigorously heat them, they are going to get either denatured, or their vitamins are going to get destroyed, or their taste is going to get destroyed. And so you cannot vigorously boil them. So Pasteur actually found a process where you actually increase the temperature of these liquid to a certain degree. Either it's a 65 or 70 degrees. We are going to talk about that later. But you increase the temperature to a certain degree, and that can avoid the spoilage of these material for a little while. So basically, you can increase the shelf life of these material to a certain extent if you just use this process, which is still used in many of these industries. So this is one of the name that was not in that list, but I definitely want you to remember, and it's easy to remember. Joseph Lister was one of the one who actually established the relationship between the microorganism and the disease and provide the indirect evidence from causal agent of the disease. So he is the first who actually developed this antiseptic surgery system to prevent the microorganism from entering the wounds. And his patients had fewer post-operative infections. And how you remember that Joseph Lister's name? Remember, the mouthwash Listerine. The mouthwash Listerine is named after Joseph Lister, and that will allow you to remember this name. So just a couple of different names which are really important as such. So the Robert Koch is also another scientist which established the relationship between the Bacillus anthracis or the anthrax. But that is of course, it was a monumental landmark discovery. But why you need to remember Robert Koch's name, that he actually proposed this Koch's postulate, which are still used today to establish the link between a particular microorganisms and a particular disease. So how this Robert Koch's postulates actually work, we're going to dissect that. So here, what we are looking at in the first postulate, we are looking at the microorganism must be present in every case of the disease, but absent from a healthy organism. So, for an example, what we are trying to do, that the Koch developed a staining technique to examine the human tissue, and Mycobacterium tuberculosis could be identified as the diseased tissue. So you actually take the tuberculosis from there and you can just stain them. So then in the second postulate, the suspected microorganism must be isolated and grown in a pure culture. So Koch actually grew the Mycobacterium tuberculosis in the pure culture in coagulated blood serum as well. So he actually found them in there. And the same disease must result when the isolated organism inoculated into a healthy host. So Koch injected the cells from the pure culture that he grew onto the coagulated blood serum into some guinea pigs. And they found that the guinea pigs subsequently died due to the tuberculosis from the cells which were grown on this serum. Now, the same microorganism must be isolated again from the disease host, and Koch isolated the Mycobacterium tuberculosis from the lung tissues or the blood serum of the dead guinea pigs. And when he actually stained, it looked exactly the same, and the characteristic of these microorganisms were the same that was actually the lung tissues. So this is the process, and this is a process by which we can actually determine that a particular disease is caused by a particular microorganisms. And this is still a gold standard, or this is the Koch's postulate. But there are some problems with that. There are some limitations. Some of the organisms cannot grow in pure culture. And this was a huge problem. Just giving a quick example of that, like the hepatitis viruses, hepatitis B and C viruses, they were very difficult to grow in the laboratory condition. And then finally, after many years of research, some of the liver cells could grow them. So there were really no direct evidence of that as such. So we're going to talk about that, how you get those evidence. Sometimes using the humans in completing the postulates is unethical. So think about that. Suppose you have here, Robert Koch could actually infer this because this Mycobacterium tuberculosis caused disease in the guinea pigs and it died. Suppose you isolate a microorganism for a human being which will not replicate in any of the animal. Like it will not replicate in a monkey tissue. It will not replicate in guinea pigs or mice or any of the laboratory animals. It only is very specific to human beings only. So then it is almost impossible, or you have to rely on the direct evidences, because you cannot use human beings to complete the postulate. On the other hand, nowadays, a molecular or genetic evidence may replace and overcome these limits. Just a classic example, that if you actually having a particular disease, and you know that that there is an organism causing this, but you cannot grow it in somewhere else, you can do electron microscopy, or you can have different staining techniques or molecular or genetic evidences to actually supplement the Koch postulate, and that's also another standard. So some of the organisms cannot be grown in the laboratory, and those are the limitations. So Koch's work actually led to the discovery of this agar. We are going to talk about that. This is actually a plant-derived material. It is actually from a seaweed, and the seaweed, you actually get this material. It is gelatinous, and what you do, that you actually use a petri dish, and you actually have a media with the agar, and you actually form basically a semi-solid gel-like structure on the petri dish. That is a gold standard to grow many of these microorganisms. They are called, so you can grow them in a broth culture called nutrient broth, or you can grow them on this nutrient agar media on the petri dishes. We'll talk about that somewhere else as well. And also led to the method of isolating the microorganisms. And there are some of the ideas that the Charles Chamberlain actually developed some of the porcelain bacterial filters, and Ivanovsky and Bajorink actually used these to study the tobacco mosaic viruses. So what they found, that this is an interesting part, and I want you to just remember that process, and we're going to talk about that at the later part of this module, that these were the bacterial filters, and when a contaminated broth were passed through this, the filtrate actually could not cause the disease or could not spoil the broth. So they were bacteriological filters. But Ivanovsky and Bajorink actually found something, that they determined that there are some plant had this infectious agent, the tobacco mosaic plants. When a plant actually had this disease, they actually ground up these plants and passed them to these filters. And after passing through the filter, they took the filtrate and took that filtrate and put that back onto the plant, and they found the disease were there. And this is a new agent where they discovered that they must not be bacteria, they must be something else. And that was the discovery of the viruses eventually, and which can pass through these bacterial filters, because the viruses were way smaller compared to the bacteria. Bacteria's in the range of hundreds or thousands. So these were the first evidence that the viruses can pass through the bacterial filters, and that this was also a landmark discovery. So the other different developments is that Pasteur and the Rouge discovered the incubation cultures for long time intervals between the transfer of the pathogen to lose their ability to cause disease. So even before that, we actually has a clear understanding of the disease, they were able to develop some of these vaccines by the process of attenuation. That means that when they actually grew some of these different agents of disease in different animals over the period of time, which we call as intervals between the transfer, cause the pathogens to lose their ability to cause the disease. And when the pathogens actually lose their ability to cause a disease, these called attenuation. And they actually found that these attenuated agents, if injected to a healthy human being, can cause a vaccine or prevent the disease to get into them. And that's how they developed the vaccine for chicken, cholera, anthrax, and the rabies. And many of these are attributed to this Pasteur, and they all actually were relied upon this attenuation, where they the transfer in between the intervals when they were in between the cultures attenuated these disease-causing organisms to a level that when injected to a healthy human being, although there were, of course, some risks at that point of time, but when injected to the human being will not cause a disease, but will protect them by raising antibodies against those agents, which will protect them in the future. Landmark discoveries. Then we talked about that. Jenner, remember that, used the vaccination against the smallpox. We are going to talk about that. Then these are some of the scientists who develop antitoxins against the diphtheria toxins, against some vaccines. And Mechnikov discovered the bacteria engulfing phagocytic cells in the blood. So this is probably one of the very first observation of the start of an immunology. So these phagocytic cells in the blood, which are the white blood cells, he first demonstrated that they can actually destroy the bacteria. And indeed, these were some of the cells which are found in our immune system. And we're going to talk about that, some landmark discoveries. And these are some of the microbial ecology. Some of these, they actually studied the soil microorganism, pioneered the use of enrichment culture and the selective media. And they also found that there are numerous interesting metabolic process, for example, nitrogen fixation. These are some of the bacteria which are essential for the different plant life or the biogeochemical cycle, and they are involved in many different cycles in the earth, and essential part. And we are going to talk about these in detail in the microbial ecology section. So again, there are several slides that we just saw that are involved in this last part that we just talked about in length. But again, don't get too tense, because I'm not going to ask just the numbers and the names much, but I just want you to remember just a couple of different names. For an example, I will definitely remember from here the Edward Jenner, John Snow, Pasteur, and Robert Koch. These are some of the most important ones as well. And then over the period of time, that these were some of the later development. And if you remember that I just quickly skipped over that, because we are going to talk about these in some of the different areas or different courses as such. So you really do not just read that self-explanatory material, but we will not go into the detail of asking questions about these. These are very self-explanatory. So let's go into the next part that we are going to talk about, that the microbiology encompass many subdisciplines. So let's take a look at that, what we mean by that. So you will be able to construct a concept map table drawing to illustrate the diverse nature of microbiology and support a belief that microbiology is experiencing the second golden age as such, which is basically the genome after the genome projects, the genomics, metagenomics, and all those kind of things. So it's a basic aspect of the microbiology has a basic and applied aspect. The basic aspect are concerned with the individual group of microbes, microbial physiology, genetics, molecular biology, and taxonomy. We're going to cover many of these aspects. On the other hand, we are going to also talk about a whole section or a module is related to the applied aspects of the disease, water, food, and industrial microbiology as well. So we are going to talk about these. So essentially, the molecular and genomic methods led to the second golden age of microbiology. So some of these classic discoveries are the restriction endonuclease, and we're going to talk about that in certain sections, where these are the molecular scissors where we can cut the DNA molecules wherever we want in sequence specific ways, and then we can stitch them by using some other enzymes as well. So these actually led to the cloning process, and that is a great discovery. And then the first recombinant molecule was developed, and we have the recombinant insulin molecules, means that these are the product of genetic engineering by the cloning process, and we actually have many of them are available and routinely done in every lab. DNA sequencing method, but Uzi-Sanger method, there were classical DNA sequencing method, whereas now these are benchtop, tiny, small machines which are probably, I mean, like if I'm not exaggerating, are million times probably more powerful than Sanger. So basically what happens now, these the new generation sequencing techniques, pyrosequencing and all those different latest techniques, you can just get a spoon of water from a pond, and you can probably find each and every organisms in there, bacteria, virus, protozoa, by this sequencing technique without doing any prep, literally. And these are almost unheard of in earlier days. So the DNA sequencing method actually has gone leaps and bounds, and the metagenomics is a process by which these are the techniques by which many of the new organisms are being discovered nowadays on an everyday basis. And then the bioinformatics and the genetic sequencing analysis, the computational power also have gone through the roof, and we're going to talk about them in appropriate sections. So here we are just giving you an idea about the major fields in the microbiology. These are very self-explanatory. There's no reason for me to go and read the slides. So just remember that some of the details of medical microbiology, public health microbiology, immunology, agricultural microbiology, what they deal with, and these will be pretty straightforward. We are going to talk about some of these industrial microbiologies, how the penicillin and other antibiotics, vaccines, steroid, alcohol, solvents, these all fall under the industrial microbiology. These are some of the different areas that people are interested into, and they become microbiologists and go into the industries to work in many of these industries. On the other hand, you can be a research scientist where you can work on microbial genetics, molecular biology, bioinformatics, study genetic information. These are the different areas that encompass microbiology. These are very self-explanatory as well. Just having a broad overview of that. So with that, we will just wrap up. Field with a long history from the 1600, from from Leeuwenhoek's discovery to even if we just think about that, this is kind of interesting, that probably the last agent that went into a microbiology book was the prion protein, which is a microbiological agent that was discovered in 1980 by Stanley Prusiner. But he actually discovered that, whereas the history is probably more than 400 years old. So the field continues to prove insights into a greater understanding, and we will talk about in many different aspects of that. And I think that from this chapter, we will have just a brief overview, have an overview of understanding of the chapter. This is a pretty easy chapter, although the video is pretty long, and but the most of the chapter will be pretty self-explanatory, and you will not be asked in between lines questions as such. Whatever we have covered in here, pretty much you are going to see them in your practice quizzes, and you will be able to get those questions from your quizzes in a similar format and the thing. So I think you should be good. So with that, we will just end, and that will be pretty much it. So we will go from there to the next chapter.